Welcome to all. I am Dr. M. Sathish, Assistant Professor, PG and Research Department of Microbiology, JJ College of Arts and Science, Pudukkottai. Today, we are discussing about the gene transfer technology. Introduction. Gene transfer is defined as a techniques to efficiently and stably introduce foreign genes into genome of target cells. It is the subsequent stable integrations and expressions of a foreign DNA into the genome. The direct desirable gene transfer from one organism to another genome is transferred as genetic transformation. The transferred gene is known as a transgene, and the organisms that developed after a successful gene transfer is known as transgenic. History. During the in 1917s, Rogers made it become possible in introduce exogenous DNA construct in higher eukaryotic cells in vitro. In 1990, the first approved gene therapy case in the United States took place on 14th September in 1919 at the National Institute of Health under the directions of Professor William French Anderson. In 2012, Glibera became the first gene therapy treatment designed to reverse lipoprotein lipase deficiency, a rare integration disease, a pancreatitis. It was first approved for clinical use in either Europe or the United States after the European Commission. The methods of gene transfer. DNA transfer by natural methods like conjugation, transformations, transduction, transposition, retroviral transduction, and agrobacterium mediated transfer. And another two different types of DNA transfer by artificial methods. One is physical and another one is chemical methods. The physical methods like electrophorations, electrofusions, particle bombardment, micro injections, micro laser, ultrasound mediated transfer, amplifications, and magnetofiction. Another one is chemical methods, that DNA transfer by calcium phosphate co-precipitation method, liposome mediated transfer, DNA transfer by PEG medium methods, silicon carbonate fiber mediated transfer, DEAE dextrose method, DMSO polygation, and etc. First one is conjugations. It was discovered by Joshua Lederberg and Edward Datumin in 1946 in E. coli. This process involved the transfer of DNA through a direct link between the bacterial cells into the form of proteinaceous tube known as a pilus, and the plasmid is known as a fertility factor. The plasmids determine the fertility negative to an fertility positive phenotype. In some case, however, the fertility plasmids could integrate into the bacterial chromosomes, and conjugations would result in the transfer of chromosomal genes. This process, which was used to construct the first genetic map of E. coli, was termed sextraction. Bacterial that have a fertility plasmids are referred to as fertility positive or male. Those that do not have an fertility plasmids or fertility negative of female. A conjugation event occurred when the male cell extends his sex pili and one attached to the female. This attached pilus is a temporary cytoplasmic bridge through which a replicating fertility plasmid is transferred from the male to the female. When transfer is completed, the result is two male cells. When the fertility positive plasmid is integrated within the bacterial chromosomes, the cells is called HF4E cells, which means high frequency of recombination cells. Okay, next one, transformations. Transformations, it is the direct uptake of exogenous DNA from its surrounding and take up through the cell membrane. Transformations occur naturally in some species of bacteria, but it can also be affected by artificial treatment in other species. Cells that have undergone this treatment are said to be competent. Any DNA that is not integrated into the chromosome will be degraded. Transduction. Gene transfer from a donor to a recipient by way of a bacteria watch. If the lysogenic cycle is adapted, the watch chromosome is integrated into a bacterial chromosome, where it can remain dormant for thousands of generations. The lytic cycle leads to the production of new patch particles, which are released by lysis of the host. Another one is Acrobacterium mediated transfer. The Acrobacterium tumefaciens is a soil-borne gram-negative bacteria. It invades many dicot plants when they are injured at the soil level and causes ground gall disease. This disease is associated within the presence of the tumor-inducing plasmids within the bacterial cell. Co-cultivate within the Actobacterium. Small pieces of leaf tissue placed into a culture of Acrobacterium for about 30 minutes. The explant then placed in MS medium without selected agent. Incubated explants with Acrobacterium for 2 days to allow transfer of the tDNA. Kill the Acrobacterium with a suitable antibiotic. The explants are removed from the medium and washed in cefotaxime selected for the transformed plant cells. The explants are transferred to a selective medium with cofetaxin, axin, cytokinin are used to encourage the regenerations of organogenesis. Regenerations of whole plant. The shoot can be rooted by placing them on solid medium containing a high axin to cytokinin ratio. And another one is electroporation. The electroscopic pores are induced in biological membrane by the applications of high volt of electric pulse. These pores are known as electropores, which allow the molecules, ions and water to pass from one side of the membrane to another and allow to accept exogenous DNA. The electroporation has been reported to enhance the level of gene expressions and can be used to increase efficiency of transformations or transfections of bacterial cell. It significantly improve immune responsible elicitate to DNA vaccines in both large and small animals. General applications of electrophorations, introductions of exogenous DNA into a small cell lines, plant protoplast, yeast protoplast, and bacterial protoplast. Wheat, rice, maize, tobacco have been transferred with frequently up to 1% of the method. Electrophorations of early embryo may result in the production of transgenic animals. And advantages, uh, efficient transformations, a large number of transfer cells can be obtained and the least number of cells deaths. Methods of fast and low equipment cost, does not require expertise individual. High percentage of a stable transfer can be produced. Disadvantages, difficulties associated with the regenerations of plant from protoplast. Rise of obtaining genetic variations in protoplast mediated regeneration plant. Another one is particle bombardment. It is a physical methods that use acceleratory micro projectiles to deliver DNA or other molecules into intact disease and cells. Firstly, used by Klein et al. In 1987 and Sanford et al. In 1987. GeneCon is developed to enable penetrations of the genetic materials containing a gene of interested in the cell. Another one is microinjections. The microinjections where the DNA is directly injected into plant protoplast or cells using fine tipped a glass needle or micropipette. These methods of gene transfer it is used to introduce the DNA into large cells, normally performed under a specialized optical microscope setup called a micromanipulator. The process is frequently used as a vector in genetic engineering and transgenics to insert genetic materials into a single cell. Computerized control of holding pipette, needle, microscope stage and video technology has improved the efficiency of this technique. Applications of microinjection process. This process is applicable for plant cells as well as animal cells, but more common for animal cells, and it and is ideally useful for producing transgenic animal quickly. Another one is liposome mediated gene transfer. Gene transfers mediated by liposome was first discovered Fenkler in 1980. Liposomes are microscopic vesicles developed in a laboratory environment. Each liposome in a spherical ball-like structure made up of phospholipid bilayer within a hollow central space, allowing liposomes interact directly with the cells. Such vesicle, when mixed with the cells in culture, fuse with the cell membrane and deliver DNA directly into the cytoplasm. It is a first non-viral techniques revised specifically for in vivo DNA transfer. Advantages: high degree of reproducibility, long-term stability, productions of nucleic acid degradation, low toxicity.